Thank you very much for the very kind introduction, and thank you all for being here, and the organizers for inviting me to present this work. It's a hard act to follow after Laura. I was just mesmerized actually. But I'm going to kind of stay on similar kind of theme in terms of using single cell and spatial transcriptomics or spatial biology to understand how tumors evolve in today's talk, mainly for therapy-induced evolution of cancer. All right, so fundamentally, my lab's interested in looking at cancer through an evolutionary lens, and most of the work that we've done in the past has really focused on transcriptomic and epigenetic plasticity in the context of tumor progression in metastatic and therapy-resistant disease. But as Laura was pointing out towards the end, it's really not enough to study tumor cells in isolation for many different cancers, because as you know, tumors really represent this entire ecosystem of highly interacting and interdependent cellular communities, the classical seed versus soil hypothesis. So we're really interested in defining this complexity of the microenvironment at the level of single cell and spatial resolution, and also investigate the mechanisms of these cross-regulatory interactions between these communities, and to figure out answers to questions such as how does this microenvironment, especially in the context of early chronic disease, something again that Laura pointed out, perhaps like with Pannon's, but again, we study liver and gastric cancer and gastric and colorectal cancers. So in the context of chronic disease, how does the microenvironment then drive progression to tumorigenesis? And then, of course, how does the microenvironment promote tumor progression, say metastatic relapse or therapy resistance? And for purpose of today's talk, mainly focus on how the microenvironment might modulate therapy response, especially in the context of immunotherapy. All right, and the way we do it, well, we serve as modern-day cartographers, so we're making atlases or maps of tumors, in this case, using single-cell approaches, which allows us to dissect heterogeneity, identify rare cell types, and then spatial biology, which then allows us to identify the geospatial localization of these tumor-specific cell types as well as the microenvironment to try to better understand what are the kind of regulatory interactions that are defining these transcriptomic states. And using these methodologies, recently we published this couple papers. This is again looking at single cell and spatial mapping in HCC in human hepatocellular carcinomas in liver. And what we identified is this remarkable reactivation of fetal programs, not just in the parenchymal cells, but also in the microenvironment. And especially we find the identification or reactivation of fetal programs in the vasculature, as well as fetal programs in the immune environment. And we could, in fact, show that spatially as well. This is looking at the OMX platform for nanostring. And we can identify these regions of interest which have very similar patterns of expression. They seem to be shared between the fetal and the tumor microenvironment, whereas the adjacent normal ecosystem of a tumor is very different from what you would see here. And most of the genes that we see that are shared in this fetal and the tumor ecosystem have telltale marks of immune suppression or immune tolerant environment. In terms of mechanisms, we identified multiple signaling pathways that we're looking at, including Wnt, VEGF, and Notch. But one thing I'm going to focus on is really VEGF, where we showed that proliferating hepatocytes, but now in fact has been shown by other cell types as well, including macrophages and some of the MDSCs or neutrophils can secrete VEGF, which then we think actually fetalizes these endothelial cells. And these fetal-like endothelial cells then can recruit and reprogram blood monocytes, and via Notch signaling convert them into immunosuppressive macrophages, which then are involved in the establishment of immune suppression. So a little deeper into what VEGF may be doing, and this is something that is more than a couple and a half decades old, that VEGF signaling, in fact, it can drive vascular remodeling, and much of it is like reactivating these fetal-like programs, which are also involved in neoangiogenesis and capillogenesis. But it promotes this phenomena called endothelial anergy, which is fundamentally that when you have these pro-angiogenic cytokines, including VEGF, they can actually block many of these pro-inflammatory signatures or signals that would otherwise actually activate expression of these endothelial cell adhesion molecules that are involved in T cell and immune cell recruitment. So once you have increased VEGF signaling, you actually block these pro-inflammatory signatures, which then result in the downregulation of many of the ICANS, VCAM, selectin, etc., many of these endothelial cell adhesion molecules. So then they no longer can recruit and reprogram immune cells, so the immune cells just will flow past, right? And this is something, again, shared between a tumor vasculature as well as the fetal vasculature. And in some ways, going back to this idea that the tumors are hijacking some of these fetal programs, which the fetus would use to evade immune surveillance, the tumors are using exactly similar mechanisms to evade immune surveillance. So then the question that we had, and something that we were very interested in, is that can anti-VEGF then normalize or redifferentiate vasculature and reverse immune tolerance and therefore restore immune response, especially in the context of immune checkpoint blockade. And so we were really lucky to get into a couple different clinical trials. I'm really going to focus on the nasopharyngeal cancer for today's talk, but we are doing exactly a very similar trial design in HCC as well. This is a trial led by our clinical collaborator, Go Boon-Cher, and the study is mainly driven by these couple brilliant people in my lab. Niranjan is a bioinformatics specialist, and Shumay is a senior staff scientist in the lab. So this is how the clinical trial was designed. So it's a two-arm study, Arm A and Arm B. Arm A is monotherapy immunotherapy alone, so just anti-PD-1 alone. Pembrolizumab, this is a collaboration with MSD. Arm B is patients, they get 1 week of bevacizumab, anti-VEGF, which is then followed by 4 weeks of a combination of pembro and bevacizumab. And what was really surprising is that the patients, the inclusion criteria was fundamentally they've basically failed all the other therapies. And what was quite surprising is that in Arm A alone, as you can see, the response rate is 12.5%. But if patients get just simply 1 week of bevacizumab, the response jumps to about fourfold, so close to 60%, with also a 3.5-fold reduction of EBV, which is a critical driver for nasopharyngeal cancer. So suggesting that simply 1 week of bevacizumab, which alone actually monotherapy has failed, right? It's part of a negative trial, but we know it can normalize endothelial cells, would somehow enhance this immune checkpoint inhibitor, right? Or immune checkpoint blockade response. The question was how. And so we had these samples, which is highlighted in these yellow time points. So we have longitudinal serial biopsies at T0 and T5 for Arm A and T0, T1, and T5 in Arm B. The clinical study has just been recently accepted in Lancet Oncology, but rest of it is a manuscript that we are submitting in terms of the translational mechanism. For those of you who are not familiar with single cells, I'm sure most of you are, each dot is representing a single cell in this cluster, right? And in these, what's called the UMAP. And every color is representing a cluster of cells with the shared transcriptome, which they basically defines their cell states. And without going into too much details, what we found is just that 1 week of anti-VEGF treatment, bevacizumab, induces quite remarkable alterations in the tumor microenvironment, including loss of Tregs and cytotoxic exhausted CD8 T cells. There are changes in the monocyte macrophages, and I'm going to come back to the molecular changes in just a bit. And obviously, there's also remarkable changes in the endothelial cells, which is the essential target of VEGF, as well as fibroblasts, right? And we can identify these cell types using these dot plots, basically, which are basically identifying all the markers that are defining these different cell types. What's the molecular nature of this altered microenvironment? What we find is that, well, firstly, we are normalizing these endothelial cells. So this is just looking at expression of many of these cell adhesion molecules. Like I said, with high VEGF, they would go down. When we block VEGF within T1, you see a remarkable activation of some of these cell adhesion molecules. We also see gain of pro-inflammatory signatures in the including this ACKR1 and IL-33. In terms of fibroblasts, there is this remarkable switch from MyCAF to ICAF. So you go from pro-tumor myofibroblastic CAFs into inflammatory CAFs, which was initially identified by David Tuvitson's lab, in fact, in pancreatic cancers models. So you see this the downregulation of MyCAF and the upregulation of ICAF markers. And at the same time, we also see gain in pro-inflammatory IL-6 positive macrophages and classical monocytes and loss of immunosuppressors. Just a little deeper dive into the ICAF states. What we find is that these emergence of within the different, so what you're looking at is a single cell UMAP now just of the cancer associated fibroblasts. And on the right-hand side, we've colored them with the response, right? CR is complete response, PR is partial response, stable disease and progressive disease. And most of these ICAFs, which are down below, these are all coming mainly from the complete and the partial responders. And you can see it in the split UMAP, right? So this particular ICAF3 population, this is 2 population, comes up in T1, then intensifies in T5. And again, if you look at the hallmarks of the genes that are highly enriched for in these ICAFs, they are all basically have these shared telltale features of an inflammatory response, suggesting that these ICAF, the MICAF to ICAF switch, may be promoting a switch in the microenvironment from an immune tolerant to an immune, and thereby somehow helping. What happens to the T cells? So this is just looking at the T cell UMAP. What we find is that, well, there is this gain in these affected CD8 effector populations, and these are the ones with granzyme and perforin. There is also a loss of Tregs, which is this pink. Yeah, just again with Bev treatment, and keep this in mind, this is all happening just with anti-VEGF, right, before adding the anti-PD-1 per se. There is loss of exhausted CD8s, and this is something that is not observed in the anti-PD-1 alone trial. If anything, you see expansion, you see activation, but a very rapid gain in exhaustion markers. FAG3 with the. So what this is telling us is that somehow this bevacizumab treatment, right, it's reversing endothelial antigen, normalizing the vasculature, which then changes this microenvironment from an immune cold to a more immune hot. And it's not observed in anti-PD-1 alone. But while the single cell analysis kind of helps us identify these cell states that are associated with the response, we don't know how these cells are organized and who's talking to who, right? So we can take these clusters and then map them into the spatial coordinates using spatial transcriptomics at single cell resolution. So that's exactly what we did with the CosMx platform, which allows us to do spatial transcriptomics at the level of single cells. And now that we start looking at the same data, but now spatially, what we find is that indeed at the baseline, you have these very disorganized endothelial cells, and which are highlighted in these yellow circles. These are the red cells, are the endothelial cells. And then just with 1 week of bevacizumab, you see this beautiful reorganization and remodeling of the vasculature. And now they're gaining these venule-like structures. And moreover, they also have expression. This is an expression of one of the ICAMs, right? And then when you look at T5, you see again, vastly reorganized and remodeled vasculature. So now the leaky capillaries are gone, but now you have this beautifully reorganized vasculature, and then you see a T cell and a B cell march. You also see some evidence of these T cell aggregates and B cell aggregates in these responder populations, something that we don't see in the non-responders. Now, instead of clustering cells based on their cell types, you can also now start clustering these cells based on common neighborhoods, right? So we can basically then cluster, and when we using some of these algorithms called Banksy in this case, and when you look at these niches that appear to be unique to responders, right? These niches basically are comprised of these different cell types, and you can then look to see which kind of cell types are then talking to each other in responders versus non-responders. And what we find is, I'm going to show you, just highlight 2 different niches. One is the niche 11, and you can see it in this light mauve or light purple here. And these are enriched for classical monocytes, inflammatory endothelial cells, pro-remodeling endothelial cells, as well as parasites. And at the same time, you also get enrichment of these niche 8, which have many of these inflammatory CD4s, ICAF3, the one that I talked about, and pro-inflammatory macrophages, IL-6 positive macrophages, as well as inflammatory neutrophils. And moreover, then you can start looking at cell chat, which is used to understand interactions or the nature of the molecular interactions within these niches. And what we find that there is indeed enhanced immunomodulatory interactions between the cell types or individual cell states that are enriched in these niches in responders, and specifically these interactions between the IL-6 positive pro-inflammatory macrophages and the ICAF3, and something that we don't really see in the non-responders. So now that we have the temporal trajectories at the level of single cell resolution, and we can map those trajectories in space, we can start basically putting together a movie of what's happening right in space in the responders versus non-responders. So this is, again, focusing on the responders. At baseline, we find generally an immune-tolerant microenvironment with dysfunctional and immature endothelial cells, disorganized vasculature, leaky vasculature. There are very dispersed parasites. There is generally a complete absence of the inflammatory CAFs, and there is presence of these immunosuppressive macrophages, them in yellow, and the absence of any classical monocytes. There's also presence of the regulatory T cells, which are the light pink cells. What happens with 1 week of bevacizumab treatment, you see this remarkable reorganization, normalization of the endothelial cells, and associated with these normalized endothelial cells, you see these blue cells coming together with these normalized endothelial cells. These turn out to be these RGS5 parasites, which are known to wrap around blood vessels to stabilize them and plug in any leaky vessels. And then you find these deep green classical monocytes coming in, which then intravasate into the tumor, and we think are then reprogrammed into these IL-6 positive macrophages. I'm not showing you the data, but with single cell trajectory analysis and RNA velocity, we can identify actually where these IL-6 macrophages are coming from with lineage analysis and pseudotime trajectory. These macrophages, the inflammatory macrophages, are always co-localized in these triads with classical monocytes and the ICAF3s. And we can show that with specific interactions between these IL-6 positive macrophages and MyCAFs, there is a MyCAF to ICAF switch. So we think these macrophages are the ones that are responsible for the emergence of the ICAFs, which then can secrete a whole bunch of chemokines and cytokines, recruit the T cells, affect the CD8s and CD4s come in. Also, a lot of B cell infiltration, which is associated with the super responders in this trial. All right, so just to conclude then, what I've talked about is that as you go from normal to tumor, you have this remarkable remodeling of the tumor vasculature into a more fetal-like leaky vasculature, which has these telltale signs of endothelial anergy. And we think that some of the mechanisms that we've identified can then invoke how we can go back and repurpose some of the drugs that may have failed as monotherapy, but now they can be combined with immunotherapy to elicit an enhanced response. And in this case, with anti-VEGF or anti-angiogenic drugs, we can then reverse endothelial anergy, or reverse immune tolerance, and then restore immune response. And with the spatial analysis, now we can combined with the spatial analysis, we can identify these niches of cross-regulatory interactions and also identify specific ligand receptor pair, which could also serve as potential therapies or therapeutics. One thing I haven't talked about is something with WNTs as well. This is turning out to be really interesting in multiple different cancers, and activated WNT signaling or dysregulated WNT signaling is also known to promote immune tolerance. And one of the ways of doing that is also by activating VEGF signaling downstream. So with that, I'll just conclude my talk here and acknowledge really these are the superheroes in the lab who do all the work. This is actually, we were climbing a volcano recently in India, Mount Ijen. And all the work that had been done by Shu, Niranjan, Shruti, Garan, and Kakali of what I've talked about. Patients, of course, without their samples, none of this would be possible. Some amazing clinical collaborators that are through, and obviously the funders who've been footing much, and happy to take.
Okay, thank you, Dr. DasGupta. So it's open for any questions, comment from the floor. Yes, Dr. Lee.
Yeah, it's fantastic work. I'm highly appreciate. So just a short question is, we know that this anti-genesis drug is block the endothelial cell, right? So as you say, so how did you avoid the hypoxia reasons after the anti-VEGF therapy to restore your immune?
Right. No. So this is a really good question. So this is kind of a factor of being at the right place at the right time. So like I said, the bevacizumab trial was actually a negative trial, and Booncher has been working. So our medical oncologist who led the investigator-initiated trial, so he's been working on bevacizumab for a while. And then what they had seen is that even though alone it doesn't really have much of a response, they had dialed down the doses of bevacizumab in a way that it promotes normalization, but there is no vascular collapse, right? And we basically built the combinatorial trial based on the previous observations, which actually has been published in Cancer Research. So we already knew the right dosing and what it's doing. So it's normalization rather than a vascular collapse. And that's a really key factor, right, in terms of anti-VEGF. Don't want to, in fact, just promote vascular collapse, because then you will get all kinds of.
That's your question. So you adjust the dose of this drug, right? So this observed in the animal model or also in the
no, that was all done with a patient trial.
Patient.
Yeah.
Okay.
That's the reason, like I said, it was part of being at the right place at the right time.
Okay.
We built it on the basis of a previous clinical trial, which was a negative trial.
So there's a vessel normalization. Okay.
Yeah,
Thank you very much.
Yeah.
Okay, Professor T, yes, please.
So I noticed that in your slide, you actually talk about PPAR alpha.
Yes.
So I was just curious about it, why you marked there, and how is also affect epithelial cell or cancer cell or other things beyond the endothelial cell in your.
Yeah, it's again a factor of giving too many talks in the last like several it's part of a different story. So we find that this is very typical in liver cancers as well, that we find that there is macrophages, especially associated with fatty liver, as well as damaged liver, where we have PPAR alpha activated. And these PPAR alpha macrophages can also produce a lot of VEGF and produce and cause this the remodeling of the endothelial cells. So the VEGF is coming not just from proliferating hepatocytes, but it could be coming from many of the myeloid cells as well. And that's something that we are on a separate project. We are trying to look to see if we can block some of those signatures or signaling pathways, especially with PPARs, to reverse endothelial angy. We're happy to discuss it offline. Okay.
Okay, one last question from me. So you have shown the alteration of the cells in those responder. Have you have chance to see those non-responder?
Yes. Yeah. So sorry. I've just in the interest of time really showed what's happening in the responders. In the non-responders, basically, well, it's not that. So okay, 60% of the ones that respond in arm B, there's still about 40% that don't respond. And in those 40% that don't respond, what we find is that it's not that, okay, well, there's about 50% of those, about 20% where we don't see bevacizumab engage, right? You don't see the normalization of the endothelium. But there is about 20% of the patients, you do see the normalization of the endothelial cells, but we do not see good recruitment of the classical monocytes macrophages, and this ICAF3 population is almost completely missing. Now, it is possible that there are poised states amongst the MyCAFs, right, that can switch, that have the ability and the plasticity to switch to ICAFs. And those are the things that are, and at least this is again purely by association, but it seems to not happen. So yeah, I mean, at this point, we don't have functional data because this is all in patients, but at least associated.
Okay, we are running out of time. Thank you again for your very wonderful speech.